So good afternoon, everyone. Let me share my screen and I'll make this a little bit bigger. I can figure out how to do that. Yes. All right, can everyone see this okay? Looks good, okay. And so I started this code. So we're gonna go over RNA visualizations today. So we're going to produce lots of different plots. And we'll do this on the airway data set, which you've seen before. And I think you have probably already most of the packages installed. I think these 3 packages may be new. So if you want to go ahead and install those on your system, that'd be great before we go into the actual code. And then just as a quick place where you can find more information about what we're doing today, I put this link here. And so most of the examples that we're going to go through today actually come from the manual. This manual is written by some of the same folks that have developed the DESeq2 package. And so they give a lot of background information about why they do certain things, why they plot things a certain way. And there's a lot more information on this website than what I have time to go over today. So I definitely would advise you to visit the website and have a look through all the information that they provide. All right, and so the first couple steps that we're going to go through are exactly the same as what Dr. Chen went over last week, which is to load the airway data set and to make a DDS or a DESeq2 data set out of that object. So, oh, I'm sorry. Oh, that's interesting. I guess I lost all my libraries. Okay, here we go. And so I'm not going to spend any time really going over this. The only thing that I want to briefly mention or briefly have a look at the data set and what's inside this data set, because you probably remember from last week that this is a data set of paired data where these researchers used 4 different cell lines and performed RNAseq on both an untreated cell line and the cell line that was treated with dexamethasone in this case. So there are 2 columns that are important here. So the cell line is in the cell column, and then the treatment status is in the dex column. And so those are the 2 that we use in the design of our DESeq data set right here. Okay, so just remember that, because we'll come back to this later when we do some of our visualizations. All right, so now the first thing we'll do is just plot a sample to sample matrix, or sorry, a sample to sample heat map to look at how similar or how different our different samples are. And because at this point we're still working with the raw count data, so nothing has been normalized yet, we're going to plot this heat map based on Poisson distances, because raw data has the distribution of raw data is better represented by a Poisson distribution than so it's not a normal distribution. So you need to use Poisson distance for non-normalized data. Once it has been normalized, you're better off using a Euclidean distance heat map. All right, so what we'll do is we extract the count data from our DDS object and transpose it, and then calculate the Poisson distance. That's all in this first line. And then we build the matrix out of that. And I'm not going to go into too much detail about exactly what we're doing in every single step, but you can, I guess by now you guys should be able to more or less follow along what's happening here. We're creating the row names. We are deleting the column names. We're setting the colors for the heat map. And then with the final one, we're plotting it. And as you can see, this heat map really nicely separates the untreated samples from the treated samples, which is great because that's exactly the difference that, or at least I'm guessing that's what the researchers were looking for, the difference in gene expression between untreated and treated samples. So this looks great, and it's a very good first check to see if your data is going to give you the answers to the questions that you were asking, even before you go into the differential gene expression analysis. All right. Are there any questions up until this point? Am I going too fast, or is this okay? Just interrupt if anyone's struggling. I don't mind slowing down. But it's hard to judge because I cannot see anyone's face. All right, we'll just slowly move on. And so again, this is exactly the same as what Dr. Chen did last week. So we're doing the differential gene expression analysis now. And then this is not the fastest computer, you can tell. And we're extracting the results. And so from the results table, the first thing that we can plot is the average expression versus the fold-to change. And so this is called an MA plot. I don't actually know where that name comes from, but here we go. And so in this plot, the points in blue have an adjusted p-value of less than 0.1. So this isn't necessarily all your significantly differentially expressed genes. And you can probably change the cutoff for this. I haven't actually looked that up, but this is what the default cutoff is. And what the reason I'm showing this graph is not because it's, it's all that great and fancy looking. But what's interesting about this graph is that it really nicely shows the sort of the relation between gene expression levels and being able to detect differentially expressed genes. And so what you see is that for all of the genes that have very low expression levels, like none of these genes reaches statistical significance. And so if you have a very low, like a gene with a very low expression level, you're hardly ever going to see or going to be able to measure upregulation or downregulation. Unless it's really, it goes from 0 to, to 50 or 200 or something, because it's plotting the average of your normalized count. But if it goes from, say, 5 to 10, which is a twofold enrichment, there is so much noise in measuring such low counts that it's very difficult to see, to get anything that's statistically significant. Whereas if you go to the other side, even very small changes can be statistically significant, right? So that's something to keep in mind if you're, depending on what it is that you want to see, you may not be able to see it just because of the nature of the data, okay? And by the way, in this plot, you see these little triangles at the top and at the bottom. These are pointing to data points that are outside the limits of the plot. And so I set the limits at from +4 to -4, and you can change those limits, but it will, to see all the data points, but it will make whatever is in the middle much smaller, obviously. Okay. So then next we're going to plot a histogram of all the p-values, which looks like this. And so these are all the p-values, and these are all the genes that fall into those bins. And as you can see, there are quite a few genes that are differentially expressed in these samples. This plot looks a little weird because it has sort of this bump here. We can make this look much nicer by filtering out genes that have very low expression levels. And so, like you saw in the previous plot, genes that have low expression levels will probably never reach statistical significance. And so that's all the genes that are sitting here in this weird peak. So if we filter out all the genes that have a base mean, so a mean expression level of anything less than 1, so basically the genes that are not expressed, we get a plot that looks like this, which is really much nicer. All right. So now we still don't know what is in this bin of differentially expressed genes, but at least we know that there's something there, and this is intriguing enough to continue and look at what those differentially expressed genes are. All right, so now, just like last week, we're going to add the gene annotation to the results table. And so I'm just going to quickly run through these and just check that everything worked okay. That looks good. And I know we already went over Volcano Plots, or Dr. Chen went over Volcano Plots with you last week. So I'll go through this quickly because I'm using the exact same package. I just put the code in here so that you have all the visualization code in one file. And also because there are a few things that I personally prefer about plotting Volcano Plots that Dr. Chen did not show last week, but I guess those are just personal preferences. And so let's start out with just a basic Volcano plot where we color everything that has a log2 fold change of greater than 2 or smaller than -2, and an adjusted p-value smaller than 0.05. Okay, here we go. Oh, and that looks awful because everything is really big now. I don't think I can. No, I cannot change the font on only one window. Well, you guys know what it's supposed to look like. Okay, so we can change the settings for the cutoff for the log2 fold change. So if you want to be a lot more stringent, we can do that, and we can also be much more stringent for the p-value cutoff. And then we can also change the size of the points and the size of the labels. So that's what this second version is doing. So now instead of, let's see, we're using 10 to the -5th as a cutoff for the p-value. And I'm trying to find where, oh, I actually used the same cutoff for fold change in this line. So I'm actually not changing the log2 fold change cutoff. That was a mistake. We can also change the axis limits so that we can get every single dot in the plot, or we can cut off the plot, the dots. In this case, I have to say I copied this code from a different data set, and for that data set, this axis limits made a lot more sense than for this data set. So now actually we're losing the top of the volcano. And then finally, we can modify the borders, and we can remove these grid lines. Like, I really don't like these grid lines. So we can remove that and get a much cleaner plot that looks like this. All right. So far, so good. I also am not really keeping in.
Do you mind if I ask really
Oh,
quick?
Zoom. Yes.
So if you hit the zoom button, I think that should show a nice
Can you see this?
no, I think it's just yeah, it's actually only sharing the RStudio main screen, I think.
Okay, let me just share my whole screen then. That might be easier. So now you can see it, right?
Yes.
Okay, perfect. Yeah, this looks much better. Oh, and Dr. Chen explained what the MA plot stands for. That's great. M is for magnitude, A is for average. Okay, perfect.
Just to make sure, I was wondering if you wouldn't mind explaining just really briefly, because I think we might have gone just a little bit quick through it last time. Really briefly, what this is kind of representing and how you can like if you saw this in a paper, like how would you interpret it?
Right. Well, so the volcano plot is, I think it's a great visual to really sort of at a glance look at which genes are most strongly or like most significantly differentially expressed, and both you can see both the upregulated genes and the downregulated genes. And so in one glance you see both sort of how statistically significant they are. So you see their p-value, but also how much they differ relative to the control sample. And so I guess it really depends on the data set and what you're looking for. Like in this data set, for example, I think this gene is really interesting, even though it doesn't have a super high p-value. There's a huge difference in expression levels between the untreated and the treated samples. And then there are some of the genes up here that are very statistically significant, but they don't really change all that much. Like they change maybe, what is it? Like threefold, fourfold maybe. Which is still, depending on what you're looking at, can still be biologically relevant, but it just really depends on what you're looking for. So for a gene like that with a very large fold change, what would be the most likely cause of the low p-value? Yeah, I think it's mostly just the biological variation. And in this case, it may be because they're looking at 4 different cell lines. And so maybe in one cell line, there's a larger upregulation or downregulation. I can actually, from this data, I cannot say what is up and what is down in their treated samples versus their untreated samples. I would expect maybe that if you would do 4 replicates of the same cell line, you would want to see a higher or a lower p-value for a gene like this. But there is such a thing as just biological variation. We're always working with biological systems, and there's always they're noisy, right? And so as long as they make sort of a sensible cutoff, or you set a sensible cutoff, I think you can work with genes like this. So, but yeah, so and the other thing that's sort of interesting, what you can see from this, from a plot like this, is that there's not a clear, there's not a a defined group of genes that is upregulated or a defined group of genes that's downregulated. It really depends on where you put your thresholds. And so if I much rather look at a plot like this than sort of read authors say, oh, we had 255 upregulated genes and 352 downregulated genes, because it really, like, how do you set your cutoffs? I mean, there are some general rules that we follow, but it's really, it depends on how stringent you want to be and what you consider biologically relevant. Right, and because if you move these lines around a little bit, you end up with very different numbers. So All right, any other questions about volcano plots? All right, I'm going to close this one and move on. Because the next plot we're going to do is a principal component plot, which I'm sure you've all heard of or seen. And so to plot a Pca plot, we're going to work with the normalized data. And so we're going to perform the R-log normalization and then plot this. And so within DSeq2, there is another function that's called plot Pca that will generate a nice volcano plot for you. And so, sorry, not a volcano plot, a principal component plot. And so principal component analysis sort of basically tries to project variation in the data onto sort of a 2D how do you say it? In a 2D format. And so it tries to explain the variation in the data through different components. I know this all sounds very abstract. I cannot explain this very well. I'm always struggling with this. But so in case of this data set, what you see is that you get a separation between the 4 untreated samples and the 4 treated samples. And so there is a clear difference in the data that separates those 2 groups of samples. And that's what is shown in the first principal component. So most of the variation in the data is explained by the fact that the different treatment status between these 2 samples, or so between the 2 different conditions. And so this plot also gives you how much variation in the data is explained by that. And so in this case, 39% of the variation is explained by treatment status. But that means that there's still a lot of other variation in the data that's not explained by treatment, but by something else. And so the second component is plotted on the y-axis, which shows sort of these 6 samples clustering together, and then these 2 are separated from the rest of the group. And so what's nice is that you can also, so right now I've just colored them by DEX, so by untreated versus treated, but you can also color them by both DEX and cell type. And so when we plot that, we get different colors for every single sample, and you can see that these 2 samples up here are the same cell line, either in the treated or the untreated version. And so it seems like there's something different about this cell line, expression levels in this cell line compared to the other 3 cell lines. And that's what sort of separates out on the second component. And so, there are 2 questions. Is there a general rule for when or why to pick R-log versus VST normalization? I think Dr. Chen can probably say more about this. From what I read in the manual is that VST is, sorry, I'm blanking on my English words today. It's not a good day to not be able to speak English. VST is recommended for large data sets. If you have more than 30 samples, they recommend to use VST. For slightly smaller data sets, they recommend R-log. But I don't know too much about the difference between the type of normalization. And what's actually interesting about, because I've ran both normalizations on this data set, and I've created the same sample to sample heat map that we created all the way in the beginning on the raw counts. And when you look at those plots, the plots actually look much better for the VST normalization for this data set. But I don't know if that is just sort of intrinsic of this data set, and you know, normally the R-log looked equally fine. But on the R-log data, you don't get the same separation of treated versus untreated samples, even though you do see it in the Pca plot, and that's something that I don't totally understand. But if you draw the Pca plot based on the VST data, it looks exactly the same as this one. So I don't know if you want to say anything about this, Dr. Chen.
No, the VST just stands for variance stabilization, normalization. So it's just sort of a cut the variance at the bottom end to be the same as top end. So I think it's just it's the same function that the you are using the R log. Okay, yeah. It in the way that the I think DESeq2 Michael Love specifically highly recommend to use the VST, but I don't know in normal application people still prefer just the size factor normalization and just move on. So that you may be right. I haven't seriously looked at the VST as well.
Yeah, so that I guess doesn't really answer your question, but it's there's more literature that you can explore if you're trying to decide what works best for your data. But yeah, there's not a clear answer for this. And then I saw another question is, what if the treated and untreated samples are mixed in the Pca plot? No, we don't just delete data. That's not the right way to go about it. That may mean that, maybe there is not a big difference in gene expression between your treated and untreated samples, for example, right? I mean, this is the outcome of your experiment. If you don't see clustering of groups in your Pca plot, it probably means that you don't have a very large difference in gene expression. Because if there is a large difference in gene expression, you would see the separation in your plot.
Just to pitch in one more, I see Henry is typing the VST as well. So I think Henry, you mostly think using this one for the Pca, or you don't use this for anything else though, right?
I usually use VST whenever I need like a between sample normalized read count. So like if I'm doing anything like, so like I think in this lecture, Dr. Bunnick is going to talk about how you might make like those sort of like box plots or like the dot plots where you're comparing expression on like a particular gene. But I often will I'll take out the VST counts to often do that, just because I think it's a very robust between sample normalization. But I think the geometric means that DESeq does naturally is also very good. I think they're all very good. They're all better than like TPM or something like that.
Yeah. No, I'm just saying it's used for visualization. I don't mean for the statistic test or the other thing. So,
oh no, yeah. Differentialization.
Yes. Oh yeah, because there's another comment that says, if the library size of the samples and therefore their size vectors vary widely, the R-log transformation is a better option than VST. Both options produce log2 scale data, which has been normalized by the DESeq2 method with respect to library size. So yeah, so it does depend somewhat on the data that you start with, and then to some extent it may be sort of personal preference, how you like things to look, or just what looks better. Just explore both options and see what gives a better representation of your data. Okay, so moving on. So now we can subset our genes and just extract the genes that are, statistically significant given some thresholds that we think are the right thresholds. So in this case, I'm using an adjusted p-value of smaller than 0.01. And log2 fold change of, like an absolute log2 fold change of greater than 1. and, or sorry, equal than or greater than 1. I'm actually, yes, oh, sorry. I was looking for where the absolute was. And then a mean expression level of at least 20 counts. So let's run this. So now we can look at the top and the bottom of these significantly differentially expressed genes. And so I'm going to move this up again a little bit. And I just want to mention here that when you run DESeq2 initially, it doesn't. So in this case, no, never mind. So when you run DESeq2, you give it the conditions, but DESeq2 doesn't know what is your sort of your null condition and what is your. So in this case, we have an untreated sample and then a treated sample. And basically, when you want to look at your data, you want to be able to say, these genes are upregulated in my treated sample or they're downregulated in my treated sample compared to my untreated sample. But DESeq2 doesn't really know what your null condition is. And so you have to figure out if, so in this case, we're looking at the genes with the smallest log2 fold change, which you would normally call downregulated genes. But from this table, you cannot really tell which, like in which sample these are higher and in which sample they're lower, right? So you have to just keep that in mind, that they may be upregulated, but they may also be downregulated, depending on what is your reference. And similar for the ones that have the largest log2 fold change. And so what I think DESeq2 does is it takes the conditions by alphabetical order. So in this case, I think it takes the treated first and then plots the untreated sample or calculates the untreated sample relative to the treated sample, but I'm not entirely sure if that's true. And so you can probably change this behavior if you want, but just keep in mind that, you know, upregulation and downregulation are relative terms. It depends on what you set as your reference. Okay, and so this is the tail. So these are the most, or the genes with the highest log2 fold change. Okay, so now if we want to look at sort of what individual genes look like and the expression of all the data points for like one individual gene, so we can take, for example, the gene that has the largest positive log2 fold change and plot that. And so that gives you this plot. And so we have our treated group where these where there's a low expression, and then an untreated group where there's a high expression. And this is what I meant by things are relative, because this is the gene that has the highest log2 fold change, right? So it you know, would be easy to conclude that it has the highest upregulation, but it's actually strongly downregulated in the treated group compared to the untreated group. We can also call upon individual genes. And so let's see if this still works. Yes, it does still work. Okay, I thought I forgot to change something about my code. So we can just select, you know, one of the genes from this list. So if it's not the top gene, like a little bit further down the line, we can select as well, this is my gene of interest, and I want to plot, see what that gene looks like. All right, so now we come to the first exercise that I'd like you guys to play around with, because these plots are not very pretty. You know, I don't like the legends. I don't like the fact that the treatment group comes first and then the untreated group comes second. You cannot really tell. We know that this is paired data. You cannot really tell which data points are from the same cell line. And so since you guys now know ggplot2, what I'd like you to do is replot this data using ggplot2 and make these graphs look nicer. And I'll show you 2 examples in a minute. But what we'll do is extract the gene counts using that same command. And that will give you basically a table with all the information that you need to plot this in using ggplot2. So we have the counts, the treatment, and then the cell line. And so I'll show you 2 examples of what I like this data to look like. Let me just, and I mean, there might be other, ways to visualize this, but these are 2 ways that I could come up with that at least for me, are visually appealing. One is specific for the nature of this data set, because it's a paired data set, and you can basically connect the 2 dots that come from the same cell line. And it's very clearly, very easy to see that in all cell lines, the expression of this gene goes up dramatically with treatment. And so I've given it a color, I've changed the size of the legends, and I've actually changed the color from gray to black. I've made all kinds of changes. And so I'd like you guys to see how far you can get. So these are all like little building blocks that you add on to the aesthetics of your graph. And so I just like you to play with this, and I'm happy to share the code, like the full code of how I plotted these graphs after the lecture. So you have that, and you can use that later on in your RNASeq or your ggplot2 adventures. And then on the right here is a box plot that I think is probably more relevant if you don't have paired data, but if you, for example, just have 4 replicates of the same sample. So you have 4 untreated and 4 treated samples, and you just look at sort of the average or the median expression between those 2, or between the 2 conditions. And so I'll give you, I don't know, maybe 15 minutes or so to play around with that, and then check in and see how everyone's doing. And just before I send you off on your on your ggplot2 adventure, I want to show you some of my noble failures, because I've struggled with ggplot2 at certain times where I was trying to plot things and I just couldn't get it to work. And so I want to share some examples of where things have gone terribly wrong. And so just to don't feel bad if things don't, if ggplot2 doesn't do what you want it to do. There are other people that struggle too, and I call these art with a capital R. And I think I still at some point should print some of these on canvas and put them in my office or something like that, because I really like them. And the last slide has my very favorite plot gone wrong ever, which was supposed to be a box plot, but I don't know. I probably wouldn't even be able to reproduce this. I have no idea what happened here, but it was not what I had intended. All right, I'll put this back on the 2 example plots, and let me know if you have any questions. And like I said, I'll give you about 15 minutes to play around with these.
So now, Henry, you know why I'm not liking the ggplot.
Yeah.
I do like it. I mean, when you get it to work, you get really pretty graphs, but yeah.
I know, I'm just joking.
It's definitely an uphill battle to get there, though.
It has a learning curve, though.
It definitely has a learning curve, yes, for sure.
That's the issue, yeah.
But once you have some standard code that I keep recycling the same code over and over again, and it's just a matter of, do I want a line graph, or do I want to have a box plot or a bar graph? And right. Most of the aesthetics are always the same.
Yeah, so. That's true, though.
I want to see if anyone's able to perfectly reproduce either of these. I'm going to be shooting for the first one, so see if you can get it.
Yeah, you can choose your own favorite color scheme.
Dr. Bunnick, what color palette did you use in that first one?
I just randomly chose some colors that I liked.
Oh, no. Oh, okay.
That's why I said you can choose your own color scheme or whatever, contrasting colors you like.
I used the eyedropper on the on the image, but it actually the colors like look off. So okay, thanks.
I can tell you in a second which colors I used. It's like, yeah, I don't know. I just look at that giant table with all the R colors and pick some that I like, and they don't always work out. Sometimes they look good in the table, and then I plot them, and it's like, nah,
yeah,
it's not good. Did anyone else get pretty far with one of these graphs? I'm curious to see if anyone wants to share their screen and share their progress with us, if anyone's brave enough. But if there are no volunteers, that's fine too. Like Henry said, there's a bit of a learning curve to Pca Plot too, and so it's totally fine if you didn't get that far. Oh, someone gets called out here. Simon? Expectations are high now. All right, well, like I said, I'll share the code that I made, that I used to make these two graphs with Henry, so that you can use it in all your future work. And I can just briefly show the code that went into these. Yeah, they're a little long. So what I usually do is I set a text element that I use. This just reduces some of the wording in the actual code itself. So I set it to size 14, which is what I use for the axis labels. So then this is your standard ggplot command where I read in the table and I set the X and Y and the colors and the groups, etc. And so for the first one, I'm using GUM point and GUM line. And I'm using, so line type 2 means you get a dashed line and the size is 1. You can make the line thicker or thinner, whatever you prefer. And so I always use Theme Classic because I hate that gray background that is the default for ggplot2. I don't know why they chose it, but I think it's really ugly. So I always use Theme Classic for all my plots. I just think it looks much, much better. So then here I'm setting the colors of the 4 different data points or data sets. And actually, I didn't do this for the plot that I showed you, but I also changed the name of the legend. So instead of cell with a lowercase c, it actually shows cell line, which looks better. And so these are the colors that I used. And I just sort of randomly chose them and they look nice together. In the third line, I'm setting all the text. So I'm changing the size of the legend. I'm setting so the default again for ggplot2 is to plot the axis labels in gray, like dark gray. So I'm setting it to black, which I think makes much more sense for both the x-axis and the y-axis. And then finally, I'm relabeling and reorganizing some of these things. So remember in the original plot, if you don't change this, it will plot them in alphabetical order. So it will plot treatment first and then untreated. And so with the limits, you can change the order. So I'm saying, okay, I want untreated first on the left and then treated on the right. And then I change the labels to non and dex. And then for the y-axis, I'm scaling it by log10 and I'm giving it a name. And then I'm also giving the plot a name. So that's all the code that went into that. And then at the end, I save it. And it's almost the same for the box plot, except that instead of a GOM point, I'm using the GOM box plot plus a GOM point on top of that. And I'm using the jitter to make sure that if the dots are almost at the same height, that they don't overlap, or at least they may only partially overlap with an alpha of 0.5. Okay, so that's that. And I'll send this code to Henry so he can upload it to the box after this lecture. Any questions about this? If not, we'll move on to essentially the last plot that we're going to make, which is a heat map. And so we'll make two different heat maps. We'll start with a heat map of all differentially expressed genes. And again, here we're using the normalized data to plot these. And so we extract a matrix of all the normalized counts data from our RLD objects. And then we extract the gene IDs of all our significantly differentially expressed genes. And then subset our matrix by only that set of DE genes and store that into a new object, what we call DE genes. As our annotation, we extract some data from about the conditions from our RLD object. And then we use pHeatmap to plot it. And we are clustering it by both the rows and the columns. I think so, or maybe by default, it's clustering the columns as well. I'm not sure. We have to check that. And it's scaling it by row. So let me just plot this first, and you'll see. Oh, and I see that I'm actually completely hiding the plot behind the chat. So this is what the plot looks like. And it's scaled by row, which means that we're not looking at the absolute expression levels. We're looking at the relative expression levels of all of these. Like for each gene, we're looking at the relative expression level across the 8 samples. And so you can see that for all of these genes, or essentially all of these genes, they are, more highly expressed in one set of the samples and have low expression levels in the other set of the samples. And then, like I said, these are all the differentially expressed genes. And so for approximately half the data set, you see an upregulation. And in the other half of the data set, there's a downregulation. And so, like I said, we clustered them by, or we gave the command to cluster it by row, but I think by default, it also clusters by columns because you get this really nice separation between the two treatment groups. So your untreated groups are on the left, and then the treated groups are on the right. And because we gave them all the annotation data, so both the treatment, but also the cell line, you see a second row here that has the colors of the different cell lines. And so you can actually match the blue with the blue and the green with the green. So you can see which data comes from exactly which sample. All right, so this gives a really nice overview of all the data. But there's so much data, so many genes that are differentially expressed that you cannot see for individual genes what happens. And so the second heat map that we're going to plot is just a heat map of the top 20 differentially expressed genes. And this top 20 that was just a sort of a random choice by me. Oh wait, there's a question. Why do we need to scale the data first before performing the heat map? Well, it's not before. It's part of the process of plotting the heat map. And the reason why we scale things is to be able to see, to be able to better visualize the differences in expression levels. So if you wouldn't scale by row, you would see dark red for the highly expressed genes and blue for the genes that have low expression levels. But that means that the genes that are highly expressed, say that they have 1,000 counts in the group with that where it is upregulated, and 200 in a group where it's downregulated. Those values would still be very high compared to a gene that has, say, 10 in one group and 50 in the other group, right? So if you want to look at relative expression levels and see what is up in one group and down in the other group and compare that across many different genes, you have to scale the data. Otherwise, all you're going to see is, well, these genes are highly expressed and these genes are have low expression levels, but you're not going to see the differences between the groups very well.
Yeah, if you set the where it says scale, if you set that to none, you can see what happens. It's exactly
Okay,
like Dr. Bunnick is saying.
Let's have a quick look at that. Yes. Yeah, so this doesn't look nice, right? You don't see anything. Yeah, that's a great suggestion. Can't type. Okay, so like I said, we'll plot the top 20 differentially expressed genes, and we're defining the top 20 by those genes that have the lowest adjusted p-value. You can also extract, say, the 20 genes with the largest log2 fold change or the smallest log2 fold change, and plot those in two separate heat maps. But in this case, I'm going to just go by the adjusted p-value and take that as a measure of what's most statistically significantly differentially expressed. And so the first thing we'll do is order the genes by adjusted p-value. And then because we're now only plotting 20 genes, we can actually have the gene names in the heat map, plotted in the heat map as well. That was, that information would have been completely lost in the first heat map that we generated. So I'm going to extract the ensemble data from this object, the ordered differentially expressed, significantly differentially expressed genes, and also extract the name. And then again, subset our matrix, our count matrix, using the ensemble the ensemble IDs, and replace the row names. So this matrix, I can show you, now has as a row name the ensemble IDs. We're going to replace those by the gene names. Oh, interesting. Let's change that. And so now, man, I can't type. It looks like this. So now we have the gene names on the side. Actually, there's one that doesn't have a gene name. Interesting. We'll see what happens with that. And then take the top 20 genes from that object and store that in the object that we're ultimately going to. Hang on, I've been messing around with some data here. I was trying to get this. I think this row just needs to go. So, and then plot this, and then it should look good. All right, yes, there we go. Sorry, so that one line that is in there that was at line 148, that should be removed. That was a remnant of something that I was messing around with, trying to get the gene symbols into the matrix. All right, so now we have a heat map of the top 20 differentially expressed genes, where some of them are, well, again, relatively downregulated, I guess, and most of them are upregulated relative to the, so in a treated sample relative to the untreated sample. And here on the right, we have for each row the gene name that's of the data that's shown. And again, the top 20 here is very, that was just it's very arbitrary choice. You know, depending on what you want to show, you may want to show the top 10 or the top 30 or whatever, or make a more a cutoff based on log 2 fold change or something like that. All right. So that was everything that I wanted to show. And so as a second exercise, Henry has prepared a data set from Recount 2. And that data set is, did you put this on box as well?
I just realized I didn't. Give me just 1 second. I'm so sorry about that.
It will be there in a second. And so he will show you, he's actually done some reformatting of the data, or he had to get some of the metadata from the short read archive to make sure that you would be able to do this exercise. And he can talk you through the steps that he did to get the data in the right format.
Sure. Would you like me to share my screen?
Sure.
Okay.
Yeah, I'll stop sharing mine.
Okay. So let me share this really quick. That's currently uploading, so you should have it in just a second. While that's happening, does anybody have any questions they wanted to ask? Or any comments or anything like that?
Or anyone have a question to install softwares again in a way?
Okay. So that's now uploaded. So here, let me copy the link. So I put the direct link in the chat. So you should all be able to access that. So go ahead, download that. You'll need to know where that is. So for example, if it's in your downloads, or I'm going to share my screen. All right, so this is what will come up when you do that. Can everyone see my Chrome screen here?
Yeah, I can see it.
Okay, great. So you'll hit download. That's going to go somewhere. It'll be in your downloads folder probably if you've got a Windows computer. So I'm going to take this and I'm going to put it with the, I'll just put it in my document since that's kind of easier for me right now. Because my home directory, oh, actually my home directory is my downloads. So find where your current working directory is and make sure your data is there. And if you're confused about where it is, it's right here on RStudio. So if you look here right above the console, that's where it is. You can also type get wd, and that'll also tell you. All right, so I have this in my working directory now. And when I load it, it shows up as SE. That's the name of the object. So basically, this is an R data file. It contains objects in R. And so the only object inside of the file was SE. And so when I loaded it, it loaded SE into my environment. So if I just take a look at SE, I can see here it's a range summarized experiment, just like Airway was. And just like Airway, it's got the call data. So call data of SE. So this describes the experiment. Basically, I got this from Recount 2, which is a website that we're going to talk. Actually, I can just show you it really quickly since I can show you how I got this. So I got this from Recount 2. If you go to Recount 2 in your browser, you can find it here. basically Recount 2 is a project where they reprocessed a lot of RNA sequencing data from public databases. And you can actually search here for studies that you might want to analyze yourself. In this particular case, we have this ERP, 010786. That's the name of the study that we have. And so this is that study. It's RNASeq of human epithelial cell line, LOVO, following Listeria infection. And to actually get this R data object, all you have to do, click that right there, and it'll download the R data object for you. So just like with an RDA is just a kind of a shorthand for R data. You can load both of those into R using this load command here. Now I modified that object because when I first loaded it, it didn't have this condition column. So it wasn't clear what the condition was. So I went to the actual study in SRA, which you can do if you come over here. If you just do open a new tab, you can find the study here in SRA. And if I look at the, if I look at the experiments, if I go to the, this is probably the best way to do this. If I look at this, this now allows me to see the different experiments that there were here. So I can see ERR, 556 is the last part of this. I come over here, ERR 556, that's that first one. And so I can see it's uninfected. So that's why I put uninfected here. So 7 and 8 are also uninfected. And then 59, 60, and 61 are all infected. So that's why you've got uninfected, 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 infected, infected, infected. Okay. So because the homework is going to involve you going to Recount and actually getting some data, I wanted to show you how I assigned this condition column, because that will not be there. Most likely that will not be there when you first download it. So the other issue is that you've got these row names. Now this is ensemble gene IDs, but they've got this dots here. That's a version number. So this is the gene ID, and then there's a version number. But when we actually transform the gene IDs into symbols, you have to have the gene IDs without the version number, or the database isn't going to be able to make that transition. So because when you do your select statement up here, when you're doing your select statement with the annotation DBI, the ensemble column in the database here doesn't have these version numbers. So that means that for you to use this, you have to just cut them off. And probably the easy way to do that is what I've shown here. This is called G sub. So basically G sub accepts a vector. So this is a vector of character vector. And then there's a pattern. I'm not going to explain what this means. If you want to learn about regex, I would highly recommend to do that when you get a chance. But this is a very simple regular expression, or regex. And basically saying, find this pattern. This pattern is going to find this version number. And then I'm saying replace it with nothing. So let's see what that does. Okay, great. So now I've got my just raw gene IDs. All right, so hopefully everything I've just said now is enough for you to be able to, on your own, do this for the homework. But now that you've seen this, go ahead and let's take 10 minutes, and we'll come back at the end. Let's take 10 minutes or so and see if you can go through at least some of the activity, and then we'll have a chance to answer some questions before we break. So Lead, you asked a really good question, which is what's the meaning of the regular expression? So just to put that simply, it basically means that you've got a period. So that's what's called an escape character. Backslash period means escape period. Basically, the reason we have to do that is because if we want to find a period, because period's a special character in regular expressions, we have to escape it if we want to find a literal period, because period without the escapes just means literally anything. That's what the dot means. It means literally anything in regular expression terms. Yeah, so yeah, that's what Simon says. Yep. Yeah, so actually, yeah, Simon's explanation, that's actually even more concise than I was going to be. So yeah, that's exactly right. So Simon said it's a regular expression. It defines a pattern in text to find and replace. The first, dot, escape dot specifies a dot or a period. Next, the dot plus is any character. So right, and then Dr. Bunnick said plus means one or more instances, right? So if you had done dot asterisk, that would have meant any number, including 0. But dot plus means there has to be at least one instance of any character or digit or what have you. No problem. Yeah, regular expressions are really, really cool. I would definitely recommend everyone take the time to just kind of like Google regular expressions in R. Or yeah, just in general, just because they're probably the most useful or one of the most useful, and they've got a little bit of a learning curve, but I think it's one of the most useful things you can learn in any programming language. Also, because they're not language specific, you can learn them in Python and then it'll be basically transferable almost entirely to R or to to Shell or pretty much any other language, I think.
Do you know why R has the double backslash as the escape character? Is there something about a single backslash that means something different in R? Because in Perl, it's a single backslash as an escape.
Yeah, I actually don't know. I've run into this issue when I do like web development in R, where like escaping in HTML and like when you're doing like R's sort of HTML portal, it's definitely kind of confusing. I'm not really sure why though, to be honest with you.
The backslash is taken as well, so you have to use backslash. That means it's not backslash. It's an escape.
I see.
Crazy.
So backslash by itself has a meaning in R already.
Yeah.
Okay.
Huh. Do you know what it means?
It's used. I think it's because used for the I think for the. I really think just for the directory. So they try to deal with that. I don't think it's for anything else though. But it is used. So so that's what the anything used, you have to use backslash. But it's backslash is used and then it's used. I think so. You have to look at it.
I think it's also used for like new line and tab and stuff like that.
Yeah. So it's a new line that's slash use slash t slash n thing.
Yeah.
It's used, yeah. So it's taken, the meaning is taken, then you have to take it.
Okay. So just for the sake of time, actually, Dr. Bunnick, did you want to go through the hands-on activity number 2, or do you want me to do it?
Oh, you go ahead and do it.
Okay. So hopefully you all had a chance to get at least part of the way through this one. I'm just going to show really quick here how I did it. Okay. So the first step was to load this up and then, change the row names, which we already talked about. Then to make the DESeq data set, so that's your DDS. It shows this warning message here that has to do with some duplicate row names. And that's actually probably because of what we did to the row names here. You don't have to worry about that, though. I'm pretty sure, out of 35,000 genes, it's unlikely at least 45 are going to probably be an issue. But yeah, if you were going to, go on to publish this, you'd want to probably look into this message a little bit further and make sure that there wasn't anything going on there. So we do the Poisson distance next. And notice here that for the row names for the Poisson distance, instead of doing the dex and cell, now we just have condition. Because that's the call data variable that we're doing differential expression based on. So now we get a very nice separation of our uninfected and our infected samples. So that's what you want to see. Just for the sake of time, I'm not going to do this part again, just because this took a couple minutes. But once you've got your results, you can go ahead and do your MA plot. I guess I probably did need to do that again. Oh, I see. Well, for the sake of time, pretend like that worked. All right. The reason it probably didn't work is because results is the name of our data, and results is also the name of a function. Which in the future, I should probably have just called this res instead of results, but there you have it. So we can see our nice histograms. We can do our left join to get our gene names. So we can now see that when we look at our results, we get the ensemble gene IDs, and we also get the gene symbol, and then we also get the gene descriptions here, which is it's really nice to have those. So for the volcano plot, we can just do the last one from before. That's just got the nice title and the grade lines taken off, and just a few things that make it just a little bit nicer to actually see. So if I zoom in on this, I can get kind of a nicer view. I'll actually do this so it's easier for you to see as well. Okay, so this is really nice. This is kind of what you would expect to see typically with a volcano plot. The only thing that's a little bit disconcerting, and if you understand the biology, maybe you'll be able to explain this, but there does seem to be a lot more genes that are on this side than are on this side. And when we talk about biological interpretation next time, we'll actually touch on this briefly and give you some ideas about how to maybe understand this, and maybe some of the ways that you can deal with what could potentially be an issue. The other thing is that you can see these sort of arms like this. So we'll talk about that next time as well. But for right now, we've got our volcano plot, and we can move on. So we do the R-log, and then the R-log gives us our RLD, which is going to be our transform data. We can do our Pca plot based on condition. So just like before, what's really nice about this is that we've got this really huge biological separation. So, you know, PC1, which is the difference between our infected and our uninfected, explains 97% of the variance in the data. So that's pretty good, I would say. So we get our results, significant results, and we can see that here. So I'll just look at the head of those significant results. So we can see the head with the top results shown. So these are the top underexpressed, at least, you know, relatively underexpressed. These are the top overexpressed, or at least relatively overexpressed. So we can see which genes those are. So let's say we're interested in Plec HB1. Well, if we come over here, we can, I didn't get to this part. We can get our gene counts for that gene. And all right. We can actually see the difference here, which is kind of nice. Okay. So the final steps here are to get your R-log matrix to actually get the indexes of the significant results. That allows you to get your DE gene matrix. And again, instead of cell index, now it's just going to be condition. And let's go ahead and see what that looks like. That's probably going to be a lot. That's a lot of genes, actually. Probably shouldn't have done that. That was probably a mistake. Yeah, I would say so. So for right now, because this is DE genes, there's over 4,000 differentially expressed genes. For right now, what I'm going to do is I'm just going to choose the, I'm just going to choose 100 of these to show for the sake of time. But when you, oh, I know why that happened. Never mind. So for right now, don't worry about that, I guess, that error. That's, if anyone wants to hang out after this, I'll explain that briefly. But for the sake of time, I'm going to go through this. Okay. So now we get our top differentially expressed genes. So we're going to get our top 20 here. And we can see them here. And, oh, right. Okay, I see what the issue is. All right, for right now, I'm just going to do it like this. So you can see here we've got our infected on this side and our uninfected on this side, and these are the top 20 differentially expressed genes. So that's essentially the end of class. Now I'm going to, for the people who want to hang around, I'm going to show you how to fix the error that I just saw, because there's a specific reason it happened. So if you don't want to hang around, you're free to leave at this point. But if you want to hang around and see how you fix that, I'm going to show that really quickly here. All right, so where did that error start? Well, it started here. And we can see here that we're getting this error in hclust foreign function call. Well, so first of all, let's actually take a look at our DE genes. Oh. Thanks, Dr. Bunnick. Okay. So our DE genes is our matrix of, 4,000 rows by 6 columns. And the 6 columns are our samples. The 4,000 rows are our genes that are differentially expressed. So let's take a look at the very, at just a slice of that matrix. Oops. That's right, because it's 6. So we can see here we've got our first few rows and our, in our columns here. So now let's take a look at our annotation. So something that you'll notice right off the bat is that our annotation looks a little bit strange. And the reason is because we don't have our samples as our row names like we should, and our column name is kind of weird. So there's a pretty easy way to fix this, which would just be to do, well, I can actually show you what I would personally do. So first of all, that's probably the first issue, actually. So let's take a look at it now. So that's not the issue. The issue then is that what we really need is our condition, and then we're going to do it like this. So you can set it up like this when you're using the data.frame function. As.data.frame is just going to coerce something into a data frame. But data.frame is actually the constructor for the data frame object. So as.data.frame is going to coerce something into being a data frame. But data.frame is actually going to allow you to construct a data frame from scratch. So in this case, we're going to construct the data frame based off of this vector here. This vector is our, well, I should say it's a factor actually. This vector slash factor, this is our condition. So that's going to be the condition column. And now we can construct the row names as the row names of the call data for RLD. So as you can recall, the call data for RLD contains a lot of stuff. It contains, it's got project, sample, experiment, yada, yada. But it also has the row names that we want. So those are the sample names that we want. And so if we do it like this, we'll get an annotation that has the correct condition and the correct row names. So that should work now. And in fact, it does. And so why did this, okay, just so for whoever's still here, why do you think this might have worked where it didn't work before? Does anybody have a guess? So if you want to know, all you have to do is go here to pHeatMap, to the actual function itself. And this is actually going to give you a pretty good idea. So the issue was with our annotation call, right? That's what we changed, and then it worked. So let's actually go to the usage and see what the description for that argument is. So annotation call, it's as similar to annotation row, but for columns. So, okay, fine. Let's read annotation row then. It says data frame that specifies the annotations shown on left side of the heat map. Each row defines the features for a specific row. The rows in the data and in the annotation are matched using corresponding row names. So recall before, we didn't have the right row names. But now that we do, it's able to match our condition from our annotations to the actual matrix that we're giving it. So recall that our matrix, the column names of our matrix are our samples, right? You can see that, plots. You can see that right here. This is our matrix, basically. The heat map is really just a representation of our matrix. The column names are our samples. So for our annotation, which is up here at the top, to be matched correctly, the row names of the annotation had to be these same samples. And that's how you were able to match up the annotation to the heat map. Because as I said, like the column names of the matrix, DE genes, had to match to the row names of the annotation. And if they're not, it's going to give you an error. Unfortunately, it's a kind of a cryptic error, so it's not immediately apparent that that was the issue. Okay, so, and if we come back down here and try this, now we can see that, in fact, this has also worked. So we can see we've got our condition up here as well. All right, so with that being said, the homework is to find your own study on recount 2, analyze it as we've done here, and make visualizations. So before we break, are there any final questions? Okay, well, there you have it then. So we'll see you all next week. Thanks.